Hello friends, welcome to the class on Unacademy, and in today's session we will be learning the structure of neuronal cells and neuroglial cells and what are the functions which are played by these different types of cells. So let us start with our session, and we will just wait for students to be online, and then let us see which of the students are right now. So we'll be discussing basic structure of neuronal cells here, and structure of glial cells, their functions along with the photographs. Yes, we have students. Welcome Ketki, welcome Pushpa. Hello Preet. Hello. Hello to everyone. Yes. So the chat box is open. You can ask your comments and you can give your comments in the chat box. So the first cell in the CNS is, of course, obviously the neuron. Now, which are the three important parts of a neuronal cells? Why I have chosen this topic? Because again, there was a question in NEET PG about neuronal cells. So let us draw a basic structure of a neuronal cell. A neuronal cell comprises of three important things: a cyton, an axon, and the processes which are called as dendrons. There are three basic structure of a neuron or nerve cell: the dendrons, the cyton which covers the nuclei, and the axon. Yes, very good Kitki. Cell body, axon and dendrons. Remember, definitely Surya, your request will be definitely taken care of. We'll take a class on ECG. In fact, in December month I have taken 6 distinguished class for ECG. I'll be repeating those class even in January month. So be in touch with me. You can find me always on Facebook as Vijay Gupta Physiology, and you can also find me on YouTube as Vijay Gupta Physiology. You can just search for me and you will have all the options to contact me to give your request. Now, is it really true that all the neurons should have axon? My question is for all the students in the group: does all neurons have axon? Yes, does all neurons have axons? I am unable to see my video, just hang on for a second. Yeah. For the students in the group, does all the neurons have axon here? Great. Yes. Few neurons, they don't have axon, and to be more precise, the cell in the retinal layer, or the retinal cells, not rods and cones, but the anaxonal cells, they are called as anaxonal cells. The amacrine cells are anaxonal cells which only have a cyton and the corresponding dendrons, multi-branch dendrons. So they have connections with all the cells, but the axon remains absent in them. There is no axon here. So of course, although they don't have axon, but the area where they can generate action potential is still present, and this area where they can generate action potential is of course the area known as axon hillock. Okay, axon hillock. Very good. Yes, not all the cells have axon. Very few examples are there, but yeah, they do exist in our body. What could be the maximum length of an axon among the given options? If I ask you, an axon can be of maximally what length? 3 centimeter, 30 centimeter, 100 centimeter, or 1000 centimeter. What could be the maximum length of the axon we can have in human body? Yes, any of these options can we choose? 3, 30, 100 or 1000 centimeter. Yes, an axon can be not exactly 2 meters, but yes, it's more than 1 meter. It's more than 1 meter. So you can have a answer as a 100 centimeter option. Okay. Now, what is the significance of axon hillock here? What is the significance of axon hillock? Yes, can you tell me, all the students here? 1000 centimeter becomes 10 meters actually, because 100 centimeter is 1 meter. So for Anshu and Ketki, the 1000 centimeter can't be an answer right now for human body, because you can't have human being as long as 10 meters. Human body can be as long as yes, 2 meters, that is 6 feet. So we can have an axon could be as good as 2 meters length, 2 meters length. Okay, so yeah, that's the answer. So it requires certain mathematics also, Now we are moving to the next point. Remember, the cytosol comprises of all the endoplasmic reticulum and Golgi apparatus, where the synthesis of neurotransmitters occur. The point where axon starts is called as axon hillock. My question is, what is the significance of axon hillock? Yes, the answer is the first action potential. Remember, the first action potential is generated axon hillock. Can you all give me just a moment, just a second here. Yeah, sorry. So axon hillock is an area where we can have first action potential to be generated. Now, if I draw a neuron, a bipolar neuron, say I'll move to the next slide. Here, now I have drawn two types of cell here, a Purkinje cell and a pyramidal cell. Now we just label here Purkinje cell of cerebellum has multipolar structure, which is multiple or a very high number of dendrites, but every cell has one axon. Axon can be branched. My question is, among these cells, among this area, where will you have first action potential? Is it going to be somewhere around here, somewhere around here? I will label it as A, B, C, or D. Where will we have first action potential? Yes, where will we have first action potential for people in a group? If I have labeled here this Purkinje cell of a cerebellum is a multipolar structure, so we have 4 label things here. Yes, A, B, C and D. Which point will have the first action potential? Anshu has answered it right. Yes, people, great. Yeah, the answer is B. Very good. The answer is B because it's an axon hillock. Now let us try to understand some few points. Why axon hillock has got is the area where we have maximum amount of action potential. First, it has got a highest concentration. Very good. Everybody answered B. Yes, the answer is obviously B, because axon hillock is an area where there is highest concentration of voltage gated sodium channel. Voltage gated sodium channels. First point. Second point, the axon hillock is an area where there is minimum threshold. The threshold is minimum in this area. The threshold is minimum. The threshold is minimum is an area for the axon hillock. Similarly, the pyramidal cells in the cortex. Yes, is it right? Pyramidal cells. Where do you find pyramidal cells? Giant pyramidal cells is a word we normally use. Where do we find giant pyramidal cells? Yes. Anybody can answer this point? The giant pyramidal cells, they are found where in the CNS? They are found in the cortex, and they are responsible for motor pathways or motor system or descending pathways, corticospinal tract. The motor pathways have giant pyramidal cells, and these cells they actually are present in the lamina 5 of the cortical lamellas. So they are present in the lamina 5 of the cortex, and the only 3% of the cells are giant pyramidal cells which control the overall motor activities. So they have a huge connectivity among each other to have a very well articulated and coordinated movements. Even in the giant pyramidal cells, the first action potential will be right at the axon hillock. Now, let us try to understand one more thing here. Suppose if this being a dendron, and this being the cyton of a cell, okay, and this being the axon of a cell, I am just taking a pictorial manner. This being the dendron, this being the axon, and this being the axon, sorry, and this being the cyton. So we'll just take one more thing here. Yeah, the electrical current, it travels from dendron to cyton, and from cyton the action potential is generated in the axon. So what do we call this current which is traveling from dendron to cyton? What do we call this current which travels from dendron to cyton? Yes, what do we call this current which travels from dendron to cyton? Yes, can anybody? I'll repeat again what I am asking here. As we are just drawn a pictorial manner. The dendrons, cyton and axon, they form one unit of a neuronal cell. All of us know that there is first action potential in the axon or axon hillock. Then what do we call the impulse which travels from dendron to cyton and from cyton to axon hillock? What this impulse will be known as? Yes, the answer is this impulse is known as electrotonic conduction. The word is, yeah, local current. Very good, people. But the more apt or the more better word is electrotonic conduction. Remember, electrotonic conduction is a type of graded potential which can generate an action potential, but which itself is not an action potential. I repeat again, the impulse or the current which travel from the dendron to cyton and within the cyton itself, that local current we call it as local current. Very good, the word is local current, but the more apt word is a electrotonic conduction. It's a flow of charged particles, just a diffusion of charged particles. Electrotonic conduction is a diffusion of charged particle. is a diffusion of charged particles. So electrotonic conduction is a diffusion of charged particles. So all the impulse it travels, all the impulses means what impulses? The impulses are EPSPs. All the EPSPs and IPSPs, they travel from the dendrons, areas in the dendrons, okay, because they make the connection with the previous neurons. So dendrons, they carry all these EPSPs and IPSPs, and all these EPSPs and IPSPs, they are summated, they are mathematically added or subtracted within the cyton, and the net output will determine whether you are going to have an action potential or not. This is the very beneficial thing, because the brain works in a very precise manner. All the impulses we receive on a muscle at the muscular level has to be very much precise. So our motor movements are highly precise, or you can say accurate movements. So there has to be a very good mathematical calculation available for them. So all the EPSPs and IPSPs, they are submitted within the cyton, and then they decided whether you are going to have a AP or not. So this is the advantage of having the large number of connections before giving an impulse. So a cyton, before passing on an impulse to the axon, at the cyton level it is decided that whether this AP has to be generated, or whether this AP has never not to be generated. So this is the very beautiful thing here. So these things are called as EPSPs and IPSPs. What you have on dendron, from the other neuron is either can be a EPSP or it can be a IPSP. Okay. Now one more thing I would like to ask here. Suppose this neuron is making a excitatory connection. Please look at the diagram here again. A neuron A makes a connection onto this dendron, okay, and this neuron is a GABAergic neuron, so it releases GABA. Is it possible that this neuron makes another connection, and can it release glutamate? My question is very simple. A neuron, on one point it makes an excitatory connection or inhibitory connection. On the other hand, it also makes an excitatory or inhibitory connection. Is it possible to have a neuron which makes one connection which releases GABA and another connection which releases glutamate? Is it possible to all the dear students? Yes or no? Yes or no? Answer is no. A neuron always releases only and the neuron always releases only one type of neurotransmitter. If this neuron is for GABA, then it will only release GABA at all its nerve endings. Please remember, a neuron is specific for one type of neurotransmitter. One neuron cannot release two types of neurotransmitter. For other neurotransmitter, we need to have other types of neuronal cells. So if you want a neuron for glutamate, then you need to create a other neuron. Okay, you need a other neuron which can release glutamate. So please remember this point. This principle is called as Dale's principle. Dale's principle. Please remember, it's Dale's principle, which explains that a neuron will release only one type of neurotransmitter at all of its nerve endings. Okay, moving to the next slide. Another type of neuronal cells, the olfactory cells and retinal cells. They are typical examples of bipolar cells. Bipolar cells. Both of them are bipolar cells. So one ending which is coming out and one ending which is the receiving side. So there are only two poles for a cyton. Again, I'll just label this point A, B, C, or you can say D, but for D is not to be answer, where we are going to have first action potential. In both the cases, the answer is going to be same, where we are going to have first action potential. Again, I would like to ask a very simple question here. Which area we are going to have first action potential? Thank you, Anshu. Which area we are going to have first action potential? Yes, please give me the right option. A, B, C, D. which area is going to have first action potential? In bipolar cell, Jesus, Mary, Ahmad has answered it C. People answered B. Yes, the right answer, of course, is B. Please remember, even in bipolar cell, it's an axon hillock where we are going to have first action potential. Bipolar cells are very common in retinal cells and even in the olfactory cells. So special senses, they have much more of bipolar cells. No, D can't be the answer. Definitely D is not an answer because D are dendrons. So dendrons cannot have action potentials. Okay, so if we consider these as an axon here, so B is right answer. Okay, B is the right answer. Now the next slide is going to be quite tough in a sense. I want you to guess where we are going to have first action potential, because all of you answered it right till now. B is the place, or axon hillock is the area where we are going to have first action potential. Now in the following slide, it's a pseudo unipolar. It's a unipolar, but to be more precise, it's a pseudo unipolar neuron. Pseudo unipolar neuron. Okay, and the area, this end has a sensory receptor. One end of the axon has a sensory receptor or a sensory organ, where the myelin sheath starts. Now my question is, which area will have first action potential here? Okay, now I'll label the things again as A, B, C or D, where we are going to have first action potential. Yes, other people in the group, please answer me this thing, where we are going to have first action potential. Yes, because there are 4 areas I have marked here as A, B, C and D. So where we are going to have first action potential in the following diagram. It's a pseudounipolar neuron, it's a receptor cell, it's a receptor neuron, sensory neuron. Pooja has answered D, Anshu is doubtful about D, Vipul has answered D. Yes, other students, any idea? Kitki has answered C. Any other students? Any other choices? Because see, it's quite typical here. The first action potential in this case, yes, majority of you answered it right. The answer is D. The first action potential, please remember this statement, the first action potential is generated either at axon hillock or at the first node of Ranvier. These are the 2 important points where we can have first action potential. The first node of Ranvier again has a area of highest concentration of action potential, sodium channels. Highest concentration of sodium channels, voltage gated sodium channels. So these are the important points which I want you to remember. So the answer is of course D. In pseudounipolar neuron, since the receptor generates a receptor potential, that receptor potential from the receptor potential in the receptor gets converted to action potential in the first node of Ranvier in the pseudounipolar neuron, and it is carried to the cortex. Moving to the next part now, the glial cells. There are 6 types of glial cells. 4 glial cells are associated with the CNS and 2 glial cells are associated with the PNS. Let us look at them one by one. Before moving further, I would like to announce here, I have one more session at 7:30 on the clinical cases in physiology on my Unacademy platform, So I invite all of you to join me at 7:30 again for Unacademy special course. You can download Unacademy app and you can watch me live again at 7:30 for clinical cases in physiology. There are almost 4 to 5 cases which I am going to discuss, clinical cases, because the recent trends is about more of clinical cases rather than just simple questions. So yeah, moving to the next slide. Astrocytes. There are 4 CNS. There are 4 CNS neurons or glial cells, and there are 2 PNS glial cells. Astrocytes belong to CNS. The word aster means having many branches like a star shape structure. So astrocytes have a star shaped structure which has got many processes, and all those processes they make connection either with the blood capillaries or with the axonal cells or with the axonal or the neuronal cells. So what is the advantage? The astrocytes, they help in maintenance of today at 7:30 today. Yes, at 7:30 today. Sorry, 8:30 today at 8:30. I just missed out the time. It's 8:30 today. 7:30 to 8 was my YouTube channel, and 8:30 to 9:30 is my the star-shaped structure. So they make connections. They make connections with all the structures in the CNS, and they help in maintenance of stability, scaffolding. They help in maintenance of scaffolding also. The beautiful thing about glial cells is that glial cells are too many in number because they are smaller as compared to the neuronal cells. So they are the ratio of CNS is central nervous system. Macrophage of CNS, no. Astrocytes are not macrophage of CNS. The next cell, that is macroglia, is going to be the macrophage of CNS. Astrocytes, they maintain more about the stability of the neuronal and blood vessels. They keep the neuronal cells near to their source of nutrition, that is blood vessels. So they keep every cell in its position so as to have better connections and better type of transmission of impulses. Also, astrocytes help in synaptic connections. So they help in synaptic connections. They are electrolyte balance. They maintain electrolyte balance. So you can say homeostatically they are very important. They take any of the extra potassium or calcium ion. So they just take in any extra potassium or calcium ion and maintain the electrolyte environment of the interstitial area of the cells. Third, they also help in neurotransmission. They also help in neurotransmission. In fact, they are so effective that they have started. They have been hypothesized that the astrocytes have also played. They also play a role in the transmission of impulses. So they have now activity. They have the activity is seen also in impulse conduction. Although not fully, but yes, they have to some extent. They have been helping neuronal cells to transmit an impulse. So they are supporting cells. They are the best supporting or best friend a neuronal cells can have is an astrocytes. Okay, so they are the most abundant cells in the CNS. All the glial cells are in the ratio of 10:1. Means for every one neuron we have 10 glial cells. And remember one more thing here, that almost 50% of volume of CNS, yes, they also help in blood-brain barrier, as you can see here. They have also help in maintenance of a blood-brain barrier. They don't allow. There are one more type of cells which also help in blood-brain barrier, and those are ependymal cells. We are just going to look at them here. The second type of cells is microglia. Microglia are similar to the astrocytes, but they don't make a connection which is like astrocyte. They just make a touch contact. They are a touch contacts. They have a touch contact with the all neuronal cells, and yes, they keep on checking the health of the cells, health of the neuronal cells. Any type of dead or damaged neuronal cells, or any type of infectious material. Remember, the microglial cells, they turn into macrophage. The macrophage turn into macrophage, and then they just take in all the dead cells of the cell. So they are called as scavengers. They are more of the scavengers of the, yes, macrophage type of cell. So microglial cells, they turn into 50% is that, although the ratio, I will answer your question, Ketki. Although the ratio of glial cells to neuronal cells is 10:1, means they have 10 glial cells to 1 neuron cells. But when you consider volume of the cells in the neurons, 50% of area of the CNS is for glial cells and 50% of area of the CNS is for neuronal cells. So although the ratio is 10:1, 10 glial cells for 1 neuronal cell, but for the volume wise they have 50-50 ratio. So I'll just mention here again with numbers, the 10:1 ratio for glial and neuronal cells, but for volume they have 50-50 ratio. I hope you got this point, Ketki. It's the ratio is 50-50 is by volume and 10:1 is by the ratio of number of the cells. Okay, moving to the next cell, the ependymal cells. The ependymal cells, they line cerebrospinal fluid. So they have two types. Either they can be a columnar cell or they can be ciliated cells. We separate the CSF from the tissue. So they separate the CSF from the tissue area and they form a wall or a separation line, you can say. They also help a role in blood-brain barrier. So they have a role in blood-brain barrier and they also play a role in the movement of the fluid. In the movement, because the cilia, the beating of the cilia to and fro, allows the fluid to move further and helps in movement of fluids. So in this way they keep the fluid circulating around the all cavities. So they line all the cavities. The point is they line all the cavities in CNS and spinal cord. Remember, all the cavities in CNS and spinal cord are lined by ependymal cells. So far now we have looked for the astrocyte, microglia and ependymal cells. Can you give me the name of the third type or fourth type of glial cells, students? We have one more glial cells left for these CNS. Which glial cell? Yes, we are done with astrocytes, we are done with microglia, we are done with ependymal cells. Can you give me the fourth type of cell? Yes. It's an interesting name, M for men. That's a nice name. Okay, so the fourth type of cell is oligodendrocyte. Please remember, the fourth type of cell is oligodendrocytes. Of course, the cells which produce a myelin sheath around the nerve fibers. And the difference here is that the oligodendrocytes, oligo means more than one. And dendrocytes means processes. Please remember, oligo means more than one and dendrocytes means processes. So they have few processes and these processes they myelin the neurons. So the difference here is that one oligodendrocyte can myelinate almost 30 neurons. This is the advantage here. So we have space saving cells. Yes, they produce myelin sheath in the CNS and they can myelinate 30 neurons at the same time. So this is the advantage of oligodendrocyte. So we have studied four types of glial cells: astrocyte, microglia, ependymal cells and oligodendrocytes. Astrocyte, microglia, ependymal cells and oligodendrocytes. Moving to the glial cells of PNS. Can you give me the glial cells of PNS, students? Can you tell me the glial cells of the PNS? Any name? Yes. Because the next slide will have the name of the glial cells of the PNS. Yes, can I have the glial cells of PNS from the students? One is the, of course, the cell which myelinate the PNS neuron. Yes. The cells which myelinate the PNS are Schwann cells and satellite cells. Remember satellite cells, these are the satellite cells. They give stability to the cyton. They give stability to the cyton. It's a very important function because they support cyton for its function. And this is a pseudo-unipolar neuron. It's a type of a pseudo-unipolar neuron or a sensory neuron. So you can see that this pseudo-unipolar neuron has a cyton. The cyton is surrounded by satellite cells. Okay, and yes, these are the Schwann cells. So Schwann cells myelinate. So each Schwann cell myelinate each of the myelin sheath, which is not the case in the CNS. In CNS, one oligodendrocyte myelinates almost 10 to 30 neurons, whereas in PNS, one Schwann cell produces one myelin sheath. So if there are this many myelin sheath, there are these many Schwann cells. So they are all nucleated Schwann cells which myelin in the PNS. Of course, they also help in regeneration of the PNS neurons. The major role is they help in regeneration of the PNS neurons. So PNS neurons, they have ability to regenerate, but CNS neurons, they don't have ability to regenerate, because in PNS neurons we have Schwann cells, and in CNS neurons we don't have Schwann cells, we have oligodendrocytes. So these are the so that's all for today's class. I wish you all very good luck, and we will see you again in the next session on YouTube, which is on 25th and 26th of this month. And at 8:30 right now, after 15 minutes, I have one more session on Unacademy, and that is about the clinical cases in physiology. So I wish you, and I see you all there. Thank you very much.